Antonio Colaprico. I'm associate scientist at the University of Miami, Florida, United States. So today I will present the tool, Narbacodar tool called Moonlight. Can you go next slide, please? So just to give you an introduction, so what the tool is dealing with driver genes and oncogene and tumor suppressor. So oncogene in general is a gene that if it's mutated, we can have increasing of and progression of the tumor, whereas a tumor suppressor genes is a gene that when it's inhibited is inhibiting the progression of the tumor. Can you go next slide, please? The reason we started to think about what we can do study tumor suppressor and oncogene that they have specific behavior. For example, they can be dual role because we found in literature, for example of genes, for instance, like NOTCH1 could be tumor suppressor in head and neck or in lung cancer, and oncogene in other tumor types like ovarian, breast, or the same gene could be oncogene or tumor suppressor in the same cancer type, but changing by stages, grades, and so on, like HDAC1 that was identified as oncogene in tumorigenesis, but oncosuppressor in tumorigenesis and oncogene in tumor maintenance. Can you go next slide, please? So there are other also genes important to do a role in literature, like the RB1 family members, where as an oncogene RB can repress H2F transcription factor controlling cell cycle G1 to phase S transition, or the tumor suppressor can inhibit apoptosis, cell senescence, and chromosomal stability. Can you go next slide, please? So what's the rationale? So we thought that we can use gene expression data and network information to identify cancer driver genes, namely tumor suppressor and oncogene. Why gene expression data? Because in general, the classical validation of tumor suppressor and oncogene is performed by modulation of expression profiles, like assay proliferation, apoptosis, invasion of cells. So I thought that why we don't use gene expression data and we use this network information where, for example, an oncogene can increase the proliferation of cells and decrease apoptosis, whereas a tumor suppressor can decrease proliferations but increase apoptosis. And why they can be important, studying dual role, because it can be potential biomarker for diagnosis, prognosis of therapy, and also possible role in resistance to the drugs. Can you go next slide, please? So what has been done is, the state of art methodology. Actually, within the TCGA, the Cancer Genome Atlas Research Network Consortium, we have curated and collated 26 tools, and this paper from Bailey et al. In the Cell 2018, and together with as a team of TCGA Pangan Atlas, we have obtained like a consensus list of 299 genes using the classical methodology like 2020, MUTSEQ, OncoDrive, FML. So the classical tools that uses, for example, mutation or copy number to identify and code driver genes. Can you go next slide, please? So we developed this Moonlight, and we published it this year at this Nature Communication paper, and I think a couple of years already the back on data package that actually use the data from TCGA, and I mean it's well known now. It's a TCGA Valinx tool that I developed, and I gave also a talk last year on Biopsy 2019 in New York, and in a workshop. I have this is the link, and then I also have this F1000 poster there. Can you go next slide, please? So, okay, how is going? What is the workflow behind? So first of all, we considered the network in the gene expression data. So we perform a differential space analysis between normal tumors to obtain candidate genes that could be differential space between normal tumors. Then we infer a network and look into the literature information and its processes. We obtained a correlation score between the gene if it's differentially expressed or if the annotation of gene apoptosis, proliferation of cells could be consistent. And then we came out with this Moonlight Zeta score. And what we can see here, for instance, are the a breast cancer application where we, for instance, we obtained an integrated other multi-omics data types where the same oncogene can we identify like amplified or the using G6-2 or the tumor suppressor we found deleted. We found also hypomethylation of the oncogene and hypermethylation of the tumor suppressor, an association of oncogene to poor prognosis, also integrated also ataxic. Can you go next slide, please? I try to to do quickly in 2 to 3 minutes. So what we discovered, what we confirmed, for instance, we have discovered inactivation of tumor suppressor in intron regions here in the figure 3, and we confirmed that was an activation of tumor suppressor in general of promoter region of oncogene in promoter region, but tumor suppressor in intron regions. And this kind of behavior was also we observed this also in the chromatin accessibility regions using ataxic data from the paper in courses at Science tool, in where we found open chromatin regions for oncogene, but open chromatin regions for tumor suppressor. And what has been, described in the other ages at Science paper 2018, it is the permeability of the tissue type to allow the genes to change role. Can you go next slide, please? So what we can see here is a circus plot where we have applied TCGA pan-cancer in 18 tumor types. The outer to inner link, we can see the core by tumor type. In green, we have oncogene. In yellow, tumor suppressor. We have annotated some mutation in purple. The line, the red are oncogene in one and tumor suppressor in another tumor type. The green are oncogene in both. Yellow is tumor suppressor in both. So the dual role are in red. And what we observed that we found unbalanced, so we predicted more oncogene than tumor suppressor. Can you go next slide, please? Where has been already used Moonlight, we had this bioarchive paper since 2 years, and then we used it in the oncogenic process paper from the TCGA pan-cancer atlas paper. So we, for instance, we identify specific differences between frame shift and nonsense mutation, and also in the stemness paper and Master tool 2018, we used Moonlight to identify oncogenic process associated to the stemness. What is the stemness index? We call it this a score where we have an oncogenic differentiation. So a lower stemness index means more differentiated and less aggressive tumor, and higher stemness index means less differentiated but more aggressive tumor. Can you go next slide? I think that's it. So I have to thanks and acknowledge all the people that contribute and help me to do this kind of work, particularly Tiago Silva, Jutta Nushman, the person from TCG Bellings, Matt Bailey, and Stephen Chen, Helena from Moonlight, and part of the CPTAC consortium where we are also working now to extend not only using gene expression data, but use also protein expression, phosphorylation, PTMs, and so on. And of course, the organizer of the background data. If you are interested, we have also our website, transbioinfolab.org at Miami. We have opening postdoc position, or if under collaboration, can write us an email. Thank you, everybody, for your question. Thank you.
Thanks so much, Antonio. That was great. So we have a question from Lei Huang. Are matched tumor normal samples required for the Moonlight analysis? Thank you.
So we didn't require that to be matched or to be, let's say, paired. Comparison, we just look at the old normal and old tumors. But it's a good point, especially when we look on, like we know, for instance, immune cell compartments, so we need to look on paired to understand the immune infiltration, then the oncogenes. But here we didn't consider the matched.
Thanks. I have a question actually about the name. Where did the name for your approach come from? I'm sorry if I missed that in your talk. Moonlight.
Sorry, can you say again? I didn't get the question.
I wondered where the name of your package came from, the Moonlight. So.
Apart that I try to be romantic, but the fact that Moonlight, there are in literature moonlightening proteins, so proteins that they can have multiple functions. So I aspire to that words. So they can have like two functions. Yeah. From moonlightening proteins. Okay, thanks.
Question from Chris. Have you used Moonlight on immune checkpoint genes that often express at high levels in cancer?
Yeah, that's a great question. We are doing now is the next step, not only looking on immune checkpoint, but also specific immune modulators. So we did a TCG Pancanatras study. We have also did this immune landscape, Thorson et al., where we have 78 immunomodulators, and definitely we have planned to do it. Thanks.
Julie, okay, you go. Julie asked, what type of pathways have been incorporated into Moonlight?
Yeah, so we have created a specific 100 biological process that we call it to be important by expert biologists. So like apoptosis, division of cells, proliferation of cells. These pathways and processes can be obtained in a table that I call a table growing that is inside the package. But definitely, as we showed in other in these studies, an end user can definitely use like the MSIG DB database from the broad and take specific collection like the hallmarks of KEGs and trying to understand these directionalities and replacing our path to biological process with well-known pathways from KEG or BioCart, Reactome, and so on.
Great. Well, thanks for a really nice talk, Antonio. Thanks for joining us so late in the evening for you as well.
So sorry for the voice, and that's okay. Thank you for this opportunity.